Hello everybody, thank you for the invitation, Covaris, to speak here. I will take you with me to back to the 2009, where we had the first cooperation with Covaris. Roche is back in the market again, and we start in 2009 with 454 sequencing and on solids from Applied Biosystems, a long time ago. But that was our first introduction with Covaris, because also the DNA needs to be sheared with a single device for single sample shearing. Later on, we start with exome sequencing as a first tier test in the diagnostics, sequencing on the solid platforms, also library prep performance manually completely with Agilent, and next to that we need to shear our DNAs. And that was time consuming with only a single S2 Covaris, so we decided to invest in a bigger system, what Eric has also showed, the Covaris E210, where we can perform 96 samples in total. But later on, we start already to think about move to genomes. In 2019, we made already the comparison between the genomes and the exomes, and here you can see our history of the numbers of exomes that we run since 2013. We start with outsourcing our diagnostics. We sent out our DNAs and we got back the data and put it in our pipeline to the bioinformatics. In 2016, we start with developing our automation workflow to do it back in our institute. We start with the rapid exomes, and we start with library prepping and sequencing, 16 samples a week, turnaround time of 15 days, and complete manually. But the number increased quite fast, what you have seen in the slide before. So we need to invest in automation, and we developed one high throughput workflow for exome sequencing. Library prep done on Hamilton systems, total capacity of 288 samples a week, also with the same turnaround times. And how we can do that in our lab. We collect our samples in a central reset desk where we get our samples in, and you can see here we do more than 25,000 samples a year. And mainly the main part is EDTA blood what comes in, and we extract DNA by ourselves. But the rest is DNA that comes in from other labs, from out in the Netherlands or from outside the Netherlands. And that's the biggest problem in our workflow. And especially the other 3% different start materials. Directly after DNA extraction, we make a quality check. We do a multiplex PCR of 55 different SNPs located in the exome, and we make a profile for each patient, and we store that. And later on in the presentation, I will come back on this profile. And then we start with Covaris. We invest in Covaris at that time because we want to have one workflow. We start for the exomes with an amount of 60 until 150 nanograms per sample, and it can come from EDTA cell lines of prenatal samples or even FFPE material. We start with DNA shearing, and we aim for base pairs of 350 base pairs per sample. And you can see here also a graph of the shearing profile after Covaris shearing. We integrated the Covaris next to a Hamilton Starlet. You can see here the picture, and on the right side there is at the moment running a video how the Covaris is performing the shearing in the instrument. So the Hamilton makes a dilution for us in the Covaris plate of a certain amount of volume and a certain amount of DNA. And the Covaris system is doing the shearing, and then the Hamilton robot picks up again the plate out of the Covaris system and brings it back to the robot. This is the special Covaris plate. He picks it up and brings it back into the device. And of course there is a lid on the plate, and also the Hamilton is able to get off the lid of the plate. And when the lid is off, we transfer the samples out of the Covaris plate to the blue plate in the front. And then we are ready for library prep. The video will show you that he can take off the lid. Most important, but otherwise you will have a crash on your system. So and when we are ready for library prep, we use the library prep for exomes with the twist chemistry a couple of years, since 2016. At the moment we have 4 library robots as capacity for exomes in the week. That is not the number that we run anymore, because we see a degree in the number of samples. This one I will skip. And afterwards we do the sequencing on an Illumina platform. At the moment, you see here a picture of a NovaSeq 6000, but we run in the meantime already our exomes on a NovaSeq X. Here you can see some results out of that run. At the moment, we run 96 samples on a 10B flow cell. And these are the results of 2024, where we aim for a first-time right of more than 96%. If it fails, there is an automatically repeat of the complete library prep and sequencing. Here are some parameters of the metrics. All the samples, the green dots are a sample, and this is data from 1 year. The vertical line is the implementation of the NovaSeq X. So you see here an increase in the coverage because the capacity of the sequencer is a bit higher and we have a little bit less samples. And the bottom one is the on-target ratio of the twist chemistry, and that's at the moment a bit above 60% and quite stable. Next to that, in this you see here the insert size. Again, the vertical line is the implementation of the NovaSeq X, and you see here a little bit of drop in the insert size, and that's also sequencing dependent. And the duplicates are stable in this way. These are some important QCs where we check our workflow for. Next to that, the extra QC is the genotyping. What we have done before, after the DNA extraction, we make the multiplex PCR of 55 SNPs. And after the exome, we extract the same 55 SNPs out of that exome and we make a comparison. If the comparison is matching completely, then we can go forward with reporting, and otherwise we need to stop. What I mentioned, we are already thinking in 2019 about the whole genome sequencing, and we start automated in 2023. First with a low number of samples, and later on with more and more samples. And you see when the introduction of the NovaSeq X was popping up, we ramp up already the short-read whole genomes. And we end up last year with a little bit more than 10,000 short-read whole genomes. Also completely automated in that part. First, we start with a library prep from Illumina, where the shearing was done by tagmentation. And later on, we step over to a mechanical fragmentation. That was also a little bit caused by the NovaSeq X, because we had some issues with the Illumina prep. With the whole genomes, we start with around the 500 nanograms in total, and we start also with the Covaris shearing. At the moment, we do only DNA out of EDTA blood, but we have already some results of different sample types, and the interest is, of course, is the insert size for that type of samples more or less the same? And I can answer that on a couple of samples. You see here on the right side the number of samples. The upper samples are the EDTA samples, and on the right there are a couple of different sample types, and you see here the coverage. And the coverage, there is a risk that the coverage will drop when you combine them on the flow cell with different material types. The insert size will be stable, so everything is above the 400 base pairs. And the uniformity, it looks quite well as well. what we have done, we have automated the Illumina kit in the first instance, and what I mentioned, when we introduced the NovaSeq X, we saw some problems. We had some low library yields, less influence on the insert size during that time, and then we decide, okay, we need to step over to a new library kit, and I like the Covaris way of shearing DNA, so we introduced the NEB kit with the Covaris shearing. In the meantime, we have already done around 18,500 samples for short-read genome sequencing, and I make a size distribution of all our samples. And on the left, on the horizontal line, you see the base pairs, and on the left side, the number of samples, and you see a nice distribution of all your samples. And we aim for 400 base pairs, and the mean is 400 base pairs in this situation. In the formal days, we did 24 samples for whole genomes on an S4 flow cell of Illumina on the NovaSeq 6000. We had a percentage of around 3% that fails in the library prep. In 2024, I have no new numbers, but we saw in 2024 also, when we step into the NovaSeq X with 64 samples on a flow cell, that the library prep was 0, based on 512 samples. I know we are not anymore on 0, but we are below the 1% fails in library prep at the moment. Also some metrics from the sequencing parameters of the genomes. And the coverage, we aim for 30x with the bottom of 25x, and the red dots in this graph are below the 25. So that's our rework at the moment, that are 1 or 2 samples on a flow cell. The bottom graph, I see the colors are changed. The red needs to be green and green needs to be red. That's the duplicate reads. That is quite stable below the 15%. Here you can see older data from 24 from the NovaSeq 6000 against on the right side of the vertical line, the data of the NovaSeq X, and you can see that the NovaSeq X has also some influence on the insert size, and at the bottom picture, but also some positive news on the Q30 value. It increased a little bit. Yeah, based on that, here you see the insert length after Covaris shearing, Covaris shearing and sequencing. That's around the 400 base pairs, so it's also nice. You have no overlap in your reads when you sequence 2 times 150 base pairs, and the Q30 is also fine. That are the short reads, exomes and genomes. But we do, as we mentioned, also long read genomes, and probably you have heard on other talks already that we do some long read genomes together with PacBio. And you think long reads, that you need to have long molecule DNAs. No, the first step is you break down your DNA. The first step is DNA shearing, and we don't do that with Covaris. We do that with tip shearing in a robot. We can pick up 96 tips on the head of a Hamilton robot, and we pipette 300 times up and down our genomic DNA and break it down to until 16 kb. And that works quite fast and quite good. We prep with 2 stations with a capacity of 192 preps a week. Also some data metrics from the PacBio workflow. The coverage is in the upper graph and the mapping quality is in the lower graph. And where we go with our diagnostics. The several years we saw already a decrease in the exomes and an increase in the short-read genomes in a quite compact time period in 3 years, an increase from 2,000 short-read genomes until 11,000 short-read genomes. Will we see that same increase in the long-read genomes? That's a question mark. We hope, that's our goal. And where we end up in 2027 we will see. Next to that, last month we had a collaboration also with Covaris to test the truCOVER whole genome kit PCR-free. We perform officially first our diagnostic with the NAB kit. We collect some data, and we did again the same library prep with the truCOVER kit from Covaris. And we did the same library prep again with some adjusted volumes of the purification. And I have some data from last Wednesday included in the presentation. And here you can see 3 bars divided over 11 different samples. So the left bar is the diagnostic yield, the middle bar is the truCOVER from Covaris for the same sample, and the right bar is the truCOVER with the adjusted volumes. And in coverage you don't see any differences. It looks in comparison equal, only one outlier, but that's probably some pipetting mistake during pooling of the complete library. Based on the insert length, there you can see when you run the complete Covaris protocol that your insert size is a little bit less in size. So the mean is, I think, 325 base pairs. There is a risk that you have some overlap in your reads when you sequence 2 times 150. That's also the reason why we adjusted a little bit the protocol in the bead volume, so that we have a little bit longer molecules, that we can have the same range as the NAB kit. And yeah, I can confirm already that with that change in the volumes that we match exactly our NAB lengths. When you look to the Q30 bases, you see also that the shorter fragments have a better Q30. Yeah, that's the explanation also that your shorter reads have a little bit of better Q30 value. So it's related on the insert size. Based on the duplicates, that's almost the same, no big differences. So based on this standard metrics after sequencing, and we didn't dive deep into the variants, I can conclude that the kit is providing the same results so far as our NAB diagnostic kit. So that was a quick overview. This is all our hardware that makes our routine possible. And these are the teams that I want to thank for all make it happen. And especially my Radboud team, of course, because there takes a lot of effort to get it up and running.